Mechanisms of phytoremediation and phytoextraction of depleted uranium. Phytoremediation and phytoextraction are promising bioremediation techniques that utilize plants to remove, stabilize, or degrade contaminants from the environment, particularly heavy metals and radioactive materials like depleted uranium. These processes are particularly valuable in restoring uranium-contaminated soils due to their cost-effectiveness and minimal environmental destruction. Phytoremediation involves various mechanisms through which plants can mitigate soil contamination. Rhizofiltration refers to the uptake of contaminants from the soil or water through plant roots. Phytostabilization is the immobilization of contaminants in the root zone, preventing their migration. Phytodegradation is the breakdown of organic pollutants by plant metabolic processes. Phytovolatilization is the release of volatile contaminants into the atmosphere through plant transpiration. Phytoextraction specifically refers to the process where plants absorb contaminants from the soil through their roots and translocate them to their aerial parts, either stems or leaves, for subsequent harvest. In the case of uranium, phytoremediation is particularly focused on the uptake and stabilization of uranium species from contaminated environments. Key mechanisms involved in phytoextraction of uranium include uranium uptake, translocation, and chelation. Uranium is primarily taken up by plants in its soluble form, typically as uranyl ions. Plants utilize similar transport mechanisms as they would for essential nutrients like calcium and magnesium. Once absorbed, uranium is transported through the xylem to different parts of the plant. Factors influencing this process include transpiration rates, which help drive nutrient movement within the plant. The use of chelating agents such as citric acid or EDTA can enhance uranium bioavailability in the soil by forming soluble complexes with uranium ions. This increases the efficiency of uptake by plants. For instance, citric acid has been shown to significantly improve uranium accumulation in certain hyperaccumulator species like Indian mustard. Compilation of plants used in environmental phytoremediation of uranium radionuclides can be found in publication by Yan et al. 2021. Some of the plants include known crops such as maize. Can supplementation promote removal of depleted uranium from the environment? Supplementation can indeed promote the removal of contaminants, including depleted uranium, from the environment, primarily through bioremediation and enhanced nutrient removal strategies. Bioremediation utilizes microorganisms to degrade pollutants, including heavy metals and radioactive materials like depleted uranium. Specific microbial strains can be supplemented to enhance their metabolic activity, leading to more effective degradation and removal of contaminants. For example, genetically modified organisms are being studied for their ability to break down complex organic compounds and heavy metals effectively. The addition of nutrients can significantly improve the efficiency of microbial processes. For instance, studies have shown that supplementing with volatile fatty acids and other organic carbon sources can enhance denitrification rates, which is crucial for nitrogen removal in wastewater treatment. This principle can be applied to depleted uranium remediation by creating an environment conducive to microbial growth that can facilitate the breakdown or immobilization of depleted uranium. Phytohormone supplementation is another option. These phytohormones stimulate plant growth and enhance nutrient absorption, which could be leveraged to increase the uptake of contaminants like depleted uranium by plants or algae. The enhanced metabolic activity leads to improved efficiency in removing various pollutants from water bodies. The effectiveness of phytoremediation can be significantly enhanced through the involvement of soil microorganisms. Soil microorganisms contribute to phytoremediation through various direct and indirect mechanisms. Plant growth promoting bacteria, known as PGPB, can stimulate plant growth by producing phytohormones such as auxins, cytokinins, and gibberellins. These hormones promote root development, increase biomass, and enhance nutrient uptake, which are crucial for plants to thrive in contaminated soils. Microorganisms help solubilize nutrients like phosphorus and nitrogen, making them more available to plants. This is particularly important in metal-contaminated soils where nutrient availability is often compromised. Microbes can directly detoxify heavy metals through processes such as bioabsorption, chelation, and transformation. For instance, certain bacteria can bind heavy metals to their cell walls or convert them to less toxic forms. This reduces metal bioavailability and toxicity to plants. Plants exude various organic compounds that can stimulate microbial activity in the rhizosphere. These compounds include sugars and amino acids that not only nourish beneficial microbes but also enhance their ability to degrade contaminants. There are several types of microorganisms that benefit phytoremediation processes. Plant growth promoting bacteria colonize plant roots and enhance growth through various mechanisms previously mentioned. Notable examples include Pseudomonas fluorescens and Bacillus cereus, which have shown effectiveness in increasing heavy metal tolerance in plants. Arbuscular mycorrhizal fungi form symbiotic relationships with plant roots, improving nutrient uptake and enhancing plant resilience against metal stresses. Siderophore-producing bacteria produce typical molecules, siderophores, that chelate iron and other metals, increasing their availability for plant uptake while simultaneously reducing metal toxicity. However, there are issues related to disposal of heavy metal or depleted uranium-enriched herb. For example, contamination. Heavy metals and depleted uranium can lead to soil and water contamination if not disposed of properly. These materials can leach into groundwater, posing risks to ecosystems and human health. Heavy metals can accumulate in the food chain, affecting wildlife and potentially leading to toxic exposure in humans who consume contaminated plants or animals. Exposure to heavy metals and depleted uranium is associated with various health risks, including neurological disorders, cancer, and reproductive issues. Proper disposal methods are crucial to minimize these risks. The long-term effects of exposure to these materials are still being studied, but there is concern about chronic health issues arising from low-level exposure over time. There is often a lack of clear regulatory frameworks governing the disposal of heavy metals and depleted uranium-enriched herb. This can lead to inconsistent practices and potential violations of environmental laws. Organizations may face significant costs associated with compliance to regulations regarding the safe disposal of hazardous materials, which can deter proper handling. While incineration can reduce the volume of waste, it may release harmful emissions if not managed correctly. Landfilling poses risks of leaching unless designed with adequate containment measures. Some heavy metals can be recycled. However, this requires specialization processes that may not be widely available or economically feasible. Disposal sites for hazardous materials often face opposition from local communities due to fears about health risks and environmental degradation. Increasing public awareness about the risks associated with heavy metals and depleted uranium can foster better community engagement in decision-making processes regarding disposal practices. Phytoextraction has implications for agricultural production, both positive and negative. Phytoextraction can improve soil quality by removing toxic metals, making it safer for crop production. By cleaning the soil, phytoextraction can lead to increased agricultural productivity. Utilizing phytoextraction aligns with sustainable agricultural practices as it reduces the need for chemical remediation methods and promotes the use of natural processes to restore land. However, there are some negative effects on agricultural production. The choice of plants used in phytoextraction can limit the types of crops that can be grown in the same area. Some plants may not be suitable for food production due to their ability to accumulate heavy metals, which could pose health risks if consumed. Implementing phytoextraction may require significant initial investment in research to identify suitable plant species and optimize growth conditions, which could be a barrier for some farmers. While phytoextraction can clean the soil, it may also alter composition over time, potentially affecting future crop production negatively if not managed carefully. There are some risks associated with phytoextraction, which include environmental risks such as soil and groundwater contamination, human health risks such as toxicity from consumed plants and long-term exposure effects, agronomic challenges such as plant selection and growth limitations, as well regulatory and public acceptance issues, which include regulatory challenges and public health concerns. Thank you for your attention.